Dear students, welcome to the EPG Pathshala. I am Dr. Gurmeet Singh, Professor of Chemistry in the University of Delhi. Today, we are going to discuss about the module Chemical Force Microscopy. Under this, we shall be talking about data analysis mainly, and this comes under the paper Surface Analytical Techniques Part 1. After going through this module, you will be able to understand various factors related to the data analysis. That is, CFM studies of the interactions between various functional groups, studies of the adhesive forces between various functional groups involved in gaseous environments or in organic solvents. Then we'll come down to adhesion forces in the liquid phases, and finally representation of these FD curves in ethanol. CFM studies of the interactions between various functional groups. Chemical force microscopy, as we write in short CFM, was used to characterize the chemical heterogeneity of the two commercially available nanofiltration and reverse osmosis membranes. CFM probes were modified with three different terminal functionalities. One, methyl, that is CH3, carboxyl, COOH, and hydroxyl OH. Chemically distinct information about the membrane surfaces was deducted based on differences in adhesion between the CFM probes and the membrane surfaces using both traditional atomic force microscopy, that is AFM force, and these measurements, and specially resolved frictional images. Contact angle, titration, and streaming potential measurements provided general information about the surface chemistry and the potential variation, which largely complemented the CFM analysis, but could not match the accuracy of CFM on the atomic level. Using CFM, it was found that both membranes were characterized as chemically heterogeneous species. Now, CFM imaging, when we talk about an increase in the friction as an AFM probe is scanned across a membrane surface, it corresponds to an increase in adhesion between the probe and the membrane surface. Because the magnitude of the measured adhesion is a function of probe and membrane surface energetics, it is possible to map the spatial distribution of membrane surface chemistry by using CFM, where both normal and lateral deflection of the AFM cantilever are measured. The normal deflection is used to measure changes in surface topography, while the lateral deflection provides simultaneous information regarding changes in the frictional or friction or surface energy. We have been talking about chemical force microscopy. This can be very conveniently used to analyze the data, and this data analysis has been given here. A number of CFM studies of the interactions between different chemical functional groups covalently linked to tips and samples have appeared in the table given below. Since our initial report of CFM was decided to be given here, adhesive forces between tip and the substitute will ultimately determine the data which is given here in the table. Here there are functional groups, and pair at the tip surface has been given, and in the second column we have monolayer and the chain length. Third column talks about the medium in which the study has been made. Then adhesion has been written here, and finally tip radius in nanometers has been given. For example, For the first one, which is an acid here, COOH and COH group, we have monolayer with a chain length of thiol SAM C16. Medium has been dry here, and the addition has been 62 units, and tip radius has also been given here. Similarly, for various compounds, for example, ethane has been used, hydrogen has been used in dry nitrogen medium, and the other data has been given here. A nominal value for similar tips provided by the manufacturer can be given in bracket, which are taken as standards. Tips were coated with 40 nanometers of gold, and addition forces normalized by the tip radius F by R have been given in terms of N by M ratio. The measured adhesive forces agree with predictions of the Johnson, Kendall, and Roberts, which is in brief called JKR theory, showing that the observed interactions can be linked directly to the surface free energy. Double layer effects on the adhesive forces have also been interpreted in terms of a modified JKR model that incorporates electrostatic interactions. A biological implementation of CFM at the nanoscale level is the unfolding of proteins with functionalized tip and surfaces. Due to the increased contact area, the tip and the surface act as anchors holding protein bundles while they separate. As uncoiling ensues, the force required jumps, indicating various stages of uncoiling. A second consideration is one that takes advantage of unique nanoscale materials properties. The high aspect of ratio of carbon nanotubes, easily greater than 1000 angstroms, is exploited to image surfaces with deep features. The use of carbon materials broadens the functionalization chemistry since there are countless routes, in fact, to chemical modification of the nanotube side walls. This can be done, for example, with diazonium salts, simple alkyls, hydrogen ones, or ozone and oxygen interfaces, and amines, of course. Multi-wall nanotubes are typically used for their rigidity. Because of their approximately planar ends, one can estimate the number of functional groups that are in contact with the substrate, knowing tube diameter and the number of walls, which helps in determining single moiety tensile properties. Certainly, this method has obvious implications in tribology as well. Measurements in gases. Several studies of functional group interactions have been conducted in dry gases and are summarized at an appropriate place. The adhesive forces between tips and samples modified with SAM that both terminate in hydrophobic groups are small or undetectable within the resolution of a reported environment. Observed adhesive forces are also small when one of the SAM surfaces terminates within hydrophobic groups and the other terminates with polar groups. In contrast, significant addition is observed when both the tip and the sample SAM surfaces terminate with hydrogen bonding groups. The relative magnitudes of the adhesive forces, as was indicated in one of the earlier slides, reflect the expected bond strengths. That is, if you take one amino acid here, this will have greater than formic acid, oxalic acid force, and this in turn will be greater than hydrazine, NH2. Because the dry gases used in these studies do not preclude absorption of water and other species on the assays. We believe that ultra-high vacuum studies will be needed to determine unambiguously the magnitude of the bare molecular interactions. We come to the ambient measurements in liquids. Capillary force is a major problem in tensile force measurements since it effectively strengthens the tip surface interactions. It is usually caused by adsorbed moisture on substrates from ambient environment. To eliminate the additional force, measurements in liquid can be conducted. With exterminated tip and substrate in liquid L, the addition to FAD is calculated by using the equation Wxlx is equal to 2yll. That is, the extra force comes from the attraction of liquid molecules to each other. This is 10 pN for ethyl alcohol, which still allows for the observation of even the weakest polar-nonpolar interaction, approximately 20 pico N. The choice of liquid is dependent on which interactions are of interest. When the solvent is immiscible with the functional group, larger than usual tip surfaces bonding exists. Therefore, organic solvents are appropriate for studying van der Waals and hydrogen bonding, while electrolytes are best for probing hydrophobic and electrostatic forces. Measurements in organic solvents are chemical force microscopy experiments carried out in organic solvents probed van der Waals and hydrogen bonding interactions, whereas those performed in electrolyte solution assessed hydrophobic and electrostatic forces. These are given in the table given below, wherein we have given functional groups, that is pair at the tip surface. This starts with ethane, then for this we have monolayer adsorption and chain length size has been given. And then we have medium in which this has been carried out, that has been ethyl hydroxide all through, and addition has been given in the next column, and finally tip radius has also been given. So from here we can analyze the characteristics of these compounds further. Representative figure of FD curves obtained in ethanol using gold-coated tips and samples that were functionalized with SMs terminating in CH3 or COH groups reveal the difference between the individual interactions. Here, a graph has been given which is a plot of force versus displacement denoted by Z given on the x-axis. Here this graph has been given for various compounds like oxalic acid and then ethane and acetic acid, etc. A representative FD curve in ethanol recorded for oxalic acid, ethane and acetic acid tip sample fractionation has been given. Tip radius is 16 nanometers in such a case. Continuum and microscopic models, which has been given in this slide. Addition data can be used to assess the energetics of the different intermolecular interactions and to estimate the absolute number of functional groups contributing to the experimentally observed forces. The assessment requires consideration of the contact deformations between tip and surface. If no molecular forces are present, the tip will separate from the sample without addition. In cases of observed addition, the JKR theory of addition mechanics can be used to assess quantitatively the above 2 points. The model assumes contact range surface forces and predicts that the pull-off forces FAD requires to separate a tip of radius r from a planar surface will be given by the relation, gives us a mathematical relation between the forces where SMT is also equivalent to some other equation. This is the thermodynamic work of addition for separating the sample and tip. Gamma Sm and gamma Tm are the surface free energies of the sample S and the tip T, respectively. In contrast with the medium M, vacuum vapor V, or liquid for L, and S, gamma St is the interfacial free energy of the contacting solid surfaces. Self-consistent calculations indicate that deformations at this scale are important, and thus could be given by FAD is equal to 2 pi r WSMT. This is a mathematical relation which one eventually works out. This formula is the well-known result of a study called DMT, Darjugin, Mueller, and Toproff theory. Is, in brief, as I said, is called DMT theory, which assumes finite range surface forces and no deformation. That is zero contact area at the moment when bonds between functional group on the tip and the sample rupture. The exact value of the numerical coefficient in equation is 3 by 2 versus 2 is beyond the experimental uncertainty. Widths of addition, force distribution, plus errors in determination of K and R. Both approaches have been used to derive surface free energies from addition data. However, even when the work of addition is given more accurately by the DMT expression, the surface profile and contact area are better described by the JKR model. Use of JKR model to correlate addition forces and surface free energy also allows the estimates of the number of interacting groups, which is a very important parameter. In this slide, this gives figures which indicate deformation of a spherical tip on a flat substrate at pull-off and corresponding force profile according to the contact mechanical model. So this has been indicated here, that what way when it is compressed, it will change the shape at the surface. So to quantify the differences and uncertainties in the adhesive interactions between different functional groups, however, it is necessary to record multiple force curves for each types of intermolecular interactions. When the solvent is chemically similar to the tip and sample terminal functional group, as example CH3 groups in the hexadecane or CH2OH group in ethanol, the forces required to separate the tip and surface are small, at times very small. The observed trend in the magnitude of the adhesive interactions between tip and sample functional groups, that is oxalate, greater than the ethane, and this is greater in turn than the acetic acid, agrees with the qualitative expectation that interactions between hydrogen bonding groups, that is COH, will be greater than between the non-hydrogen bonding groups, that is CH3. The forces observed in dry, inert gas between these and similar functional group combinations parallel are results in ethanol solvent. Histograms of the adhesive forces versus the number of times this force is observed typically exhibit Gaussian distributions and yield mean adhesive forces. Experimental uncertainty here can also be taken into account. that is of 2.3 or 0.8 or 1 or 0.4 or 0.3 to 0.2 in for interactions between the COH group or CH3 group or CH3 COH groups respectively. Since the mean values do not overlap, these chemically distinct functional groups may be differentiated by measuring the adhesion forces with a tip that terminates in a defined functionality. The observed trend in the magnitude of the adhesive interactions between tip sample functional groups, that is COH or ethane or acetic acid, agrees very well with the qualitative expectation that the interaction between hydrogen bonding groups will be greater than between the non-hydrogen bonding groups like CH3. The forces observed in dry inert gas between these and similar functional groups combination parallel are results in ethanol solvent. It gives a plot of counts versus addition for formic acid, a histogram showing the number of times that a given adhesive force was observed in repetitive measurements using COH terminated samples and tips. The histogram represents approximately 400 tip sample contacts and for one functionalized tip in ethanol. This talks about probe modification and force measurement. Modification of the AFM probes and often the samples with well-defined chemical functionalities is the key to CFM. Unless a researcher wants to go through the expense of fabricating custom probes, it can be done other way also. Although several initial attempts have been non-specific adsorption of proteins, such as BSA, to place the functionalities or functionalities on the AFM tip for further chemical modifications, the preferred current modification route goes through creation of a close-packed organic monolayer on the probe surface. For these approaches, researchers typically are where they have carried out is on silane chemistry for direct modification of the AFM probe surface or pre-coat the probe surface with a thin layer of gold and subsequently form a monolayer of alkane thiols on the gold surface. Probing entropic solvation barriers with various temperature CFM or variable temperature CFM studies. Noy and co-workers investigated the microscopic origins of these effects by measuring interactions or interaction forces between specific functional group and different solvents as a function of temperature. These experiments utilized a custom-built AFM chamber that gave researchers access to a wide range of temperatures. The binding forces could decrease as the temperature increases and the thermal functions gain more energy to break the bonds. Dynamic strength of single and multiple biological bonds. As discussed in the previous sections, attaching the interacting molecules to flexible tethers provides multiple advantages for detecting specific biological interactions. Tethers provide special separation between the specific and non-specific interactions. Certain interactions between receptors, ligands pairs, and protein-antibody interactions that provide a target interaction for multivalent immunotherapeutic drugs. However, the price advantages for these involve reduction of a nonlinear spring representing the entropic elasticity of the polymer tether in series with the chemical bond of interest. Frictional force mapping. Chemical interactions can also be used to map pre-patterned substrates with varying functionalities, scanning of a surface having varying hydrophobicity with a tip having no functional group attached would produce an image with no contact because the surface is morphologically featureless. Simple AFM operations will also prove the same, that the surface is morphologically featureless. Functionalizing a tip to be hydrophilic would cause the cantilever to bend when the tip scan across hydrophilic portions of the substrate due to strong tip-substrate interaction. This is detected by laser deflection in a position-sensitive detector, therefore producing a chemical profile imaging of the surface. Generally, a brighter area would correspond to a greater amplitude of the deflection, so stronger bonding corresponds to lighter areas of CFM image map. When the cantilever functionalization is switched such that the tip is bent when the encountering hydrophobic areas of the substrate, instead, the complementary image is observed. Frictional force responds to the amount of perpendicular load applied by the tip onto the substrate is shown in figure. Increasing tip-substrate interactions provide a steeper slope, as one would expect. Of experimental importance is the fact that contrast between different functionalities on the surface may be enhanced with an application of greater perpendicular force. Of course, this comes at the cost of a potential damage to the substrate. This has been indicated with the help of a diagram, which is the plot of frictional force versus perpendicular load.
AFM has been widely used to measure nanoscale topographic images of sample surface under environments close to systems in material and biological research. In AFM, a very sharp tip made up of silicon and silicon nitride, basically Si3 and 4, is used to probe local information on the sample surface. The tip is brought close to the sample surface and the force between tip and surface in order of 10 to power 12 to 10 to power 7 n is measured as the deflection of the micro-fabricated cantilever having the tip at its end. The tip is raster scanned over the surface using a piezoelectric device that can control the tip position with sub-nanometer precision. During raster scanning, the tip surface force is kept constant by means of feedback system to provide topographic AFM image. AFM image formation is significantly affected by the nature of the interaction force between tip and sample and by the sharpness of the AFM tip. For example, spatial resolution in AFM imaging deteriorates in the presence of strong attractive force and nanospecific adsorption of biomolecules onto a tip. Surface topographic images have been obtained by probing the interfacial repulsive force at the tip sample contact area that is independent of surface chemical composition. In ultra-high vacuum, elemental discrimination of surface atom was demonstrated using non-contact mode AFM because the tip surface force mainly reflected the short-range chemical force due to the contamination-free environment. By contrast, chemical identification of surface species in air or in solution using AFM is often challenging because of the involvement of various interactions in a major tip surface force and contamination induced changes in tip surface properties. One approach to control tip sample interaction in AFM is to tailor the surface of the AFM tip chemically with a well-defined organic thin film having a specific thermal group. The use of chemically functionalized tip for AFM imaging, which is called chemical force microscopy, enhance a specific intermolecular interaction and suppress other interfering interaction. So selectively probing surface function group of interest in the range of microscale to nanoscale. Also, CFM makes it possible to measure a variety of non-covalent chemical and biological interaction forces directly as a function of tip sample distance, which is based on the force measurement capability of AFM. In the measurements, the deflection of the cantilever, which can be converted to tip surface force, is recorded at a single point on the sample during approach and withdrawal cycle at a given speed. In the retraction part of the curve, the tip suddenly detaches from the surface, which gives the force of adhesion required to rupture the tip surface contact. Tip commonly used for CFM include oxide-coated silicone, tip modified with organoxylene, self-assembled monolayer, and gold-coated tip modified with organomercaptan. So far, in materials research, CFM has been employed for number one, determining the apparent pK of surface functional group. Number two, directly measuring single intermolecular force in the solution. And number three, investigating nanoscale heterogeneity in the chemical property on sample surface. This article discusses the application of CFM for material research, specifically number one, measurement of single interaction force for understanding host-guest complexation on the solid surface, and number two, measurements of nanoscale surface property on polymer film and monoliths.
Therefore, students, after having explained in details about CFM and the data analysis that we can probably carry out with this, let's now summarize the results of this, and I'm sure you would have learnt a lot out of this module. Interaction between chemical functionalities define outcomes of the vast majority of important events in chemistry, biology, and material science. Chemical force microscopy, a technique that uses direct chemical functionalization of atomic force microscopy probes with specific functionalities, allows researchers to investigate these important interactions directly. The basic principle of CFM, some example of its applications, and theoretical models that provide the basis for understanding the experimental results. We can emphasize application of modern kinetic theory of non-covalent interactions to the analysis of CFM data, which is pretty important in order to understand these two relations. Thank you very much.